Now, welcome to this seminar series. Today we have Arif Engin Çetin from İzmir Biomedicine and Genome Center. He's a research group leader there, and he received his BS degree in Electrical and Electronics Engineering and MS degree in Electrical and Computer Engineering, both from Koç University in Istanbul. He received his PhD degree in Electrical Engineering from Boston University. He worked, after his PhD, he worked as a postdoctoral fellow at EPFL between 2013 and 2014, and at MIT between 2014 and 2016, and then as a research scientist at Omnium in San Diego between 2016 and 2018. And since then, I think he's in Turkey at İzmir Biomedicine and Genome Center. He's pursuing research on optical biosensors integrated with microfluidic platforms. He's developing handheld biosensors for high throughput and multiplexed biosensing. Recently, he's also developing a unique label-free technique for high throughput DNA sequencing, and his seminar or his talk will also be about these research topics. Here, let's welcome him and listen his talk.
Thank you for this nice presentation and also inviting me to give you my talk in this prestigious university and this really nice department. My name is Arif Çetin. I'm a research group leader in İzmir Biomedicine and Genome Center. This center is a research center that belongs to TÜBİTAK, and we're being evaluated by TÜBİTAK and the Ministry of Industry and Development. So if after your graduation you can apply like professional job applications as a researcher. I highly recommend. We are two engineer group there, mainly focusing on optics. The main goal in our group to develop portable and low-cost optical-based biodetection systems, and our main motivation to develop those systems for national security and defense, public and global health, biology and pharmacology. The main principle we are using in our biodetection platforms is surface plasmons, and I will briefly introduce you what are those waveforms. So surface plasmons are surface waves that are propagating along an interface between a dielectric and a metal, as you see here, like air and metal, gold film, or get localized around the metal feature. So their unique properties are coming from the strong interaction between the electromagnetic field of light and the free electron oscillation of the metal. In our group, we are mainly using these periodic nano arrays fabricated along a thin gold film. So these structures are working like a filter that is allowing light transmission in a certain wavelength frequency and filter out the rest. And since these light transmission is associated with strong light localization in the nanometer scale, if a material or a biological target, such as a bacteria here, sticks on the gold surface, the electromagnetic properties of light change. So you see this change in the electromagnetic profile as a spectral shift in the visible range. And then you read these spectral shift with, for example, here is classical spectrometer. So I think the only one and the best successful commercial application of surface plasmon is the surface plasmon resonance or SPR platforms. Here, what they do is they shine a laser source with an angle that is with an incident of angle that is larger than the critical angle between prism and air, such that you excite an evanescent field here, and this evanescent field excites surface plasma on the surface of a flat gold surface. So the reason behind this mechanism is the fact that there is a momentum mismatch between surface plasma and the free space of photons. So you cannot excite surface plasma with a normal incidence, with a light source coming from free space. But there are certain problems in this mechanism because this is very angular sensitive, and also you have to use bulky and very costly optical platforms to excite such phenomena. So instead of doing that, we are using these periodic gold nano-apertures, and we can excite surface plasma even on the normal incidence. Here we overcome this momentum mismatch between surface plasma and free space photon by the grating vector of the periodic structure, and this grating vector is linearly proportional to the period of that periodic structure. So compared to the commercial platform, the optic of this system is much easier to set up. So in this figure you see the electromagnetic field distribution at the transmission maximum, and I will in the following slides I will call this spot as the EOT signal. So at the EOT signal, when you look at when you look around the rims of the nanohole, you see very tight field localization, and this tight and this large local electromagnetic fields are extend into the medium. So this configuration, this electromagnetic field configuration, increase your sensitivity to the change in the local refractive index. So here, for example, I'm introducing different refractive index of bulk solutions from like water, IPA, chlorophore, and you see as the refractive index increase, the EOT signal shift toward longer wavelengths. So this is one of the application of this system to detect viruses from medically relevant concentration. So what you do is you cover this gold surface with a protein AG. This protein AG has a high affinity to the Fc region of the virus antibody. So when you introduce this antibody to this gold surface, it sticks on the gold surface through its Fc region, so it stands like a Y shape. So the glycoprotein binding site of the viruses is available for the virus. Then you incubate the virus on the surface, then virus sticks on the gold surface through its glycoprotein. So in this figure you see the ACM image of the viruses captured on the gold surface, and the presence of these viruses results in more than 100 nanometer shift in the visible range. We also introduced the use of this platform to detect Europe's most frequently reported sexually transmitted disease, which is caused by two bacteria, chlamydia and neisseria. So in different samples you can either see chlamydia or neisseria or both of them. So we proposed this platform to detect such disease to Civis Federal Institute of Technology, and we got some like more than 250,000 franc. And then what we did is here we have a microfluidic platform which has 3 channels to repeat the test a couple of times to give more reliable sensing information. And each channel you have 3 sensor spots, and each sensor has 3 different antibody. So then you flush a urine sample, and what you see is if you have nothing in the urine sample, you see no shift in the control sensor and the control sensor and sensor coated with antibody either for chlamydia and neisseria. And if you have only chlamydia, you see a change in the chlamydia sensor. And if you have only neisseria, you see a change in the neisseria, or if you have both of them, you see the change in both sensors, chlamydia and Neisseria sensor while you see nothing in the control. So, so in the classical studies using this plasmonic nanohole arrays, the readout mechanism is used to stick on, you simply stick on the maximum transmission spot and then you monitor how it changed. But if you have a very low concentration of biological analytes, this method is not sufficient to detect, like picogram per mil type of concentration. So what I did here is, instead of looking at only spectral variations only in singular spectral location, we cumulatively monitor spectral variations in multiple spectral locations. So what I did is, I calculated the transmission response in a spectral window, and then what I did is I add up all these spectral variations in a large spectral region of interest. So compared to the classical studies where people can show like picogram per mil type of limit of detection, we could go to sub 10 nanogram per milliliter limit of detection. So this limit of detection is close to the commercial SBR platforms, where like the limit of detection is around like 100 picogram, which is about like 0.1 nanogram per mil. You can also integrate microfluidic platforms to these systems to determine real-time binding events between different protein molecules. For example, here we have a gold sensor again, and we coated protein AGN. We are flushing protein AG with a certain speed, and we look at the spectral change in the sensor to determine the real-time binding dynamics of protein AGN IgG. We could, with this post-processing technique, we show that even we can perform such experiment in sub 1 nanogram per mil limit of detection level. So in also the classical nanohole platform, people usually fabricate those structures on glass, because glass is optically transparent and also gives you a strong and robust support to this thin gold film. Like the thickness of the film is about 100 nanometers, so you need a thick material to give a strong support. But the problem here is, so you excite with this system, you excite multiple EOT signals, and the problem here is that if this guy shifts toward longer wavelength, these two guys could merge, so then that could affect the reliability of your sensing results. So what I did is I insert a high refractive index silicon nitride layer to push this guy toward the longer wavelength. As I told you before, if you increase the refractive index around the surface of the nanohole, it shifts towards longer wavelength. So that's what's going on here. So glass has 1.3 refractive index. Instead, silicon nitride has 2.3 refractive index. So this guy shifts these two modes toward longer wavelength. So if you increase the silicon nitride thickness, you completely get rid of these two modes. So you have a strong EOT signal isolated from any other surface plasma excitations. So, and we recently we have our patent granted for our invention. So at this point, I showed you how we can excite surface plasma in a very simpler optics and very close sensitivity compared to a commercial surface plasma platform. But still, this system suffers from throughput, because in each chip you need to evaluate sensors sequentially rather than simultaneously. In order to solve this problem, we replace spectrometer with a camera. So what we did here, we integrate a spectrally well-defined light source, as you see here, a laser that is spectrally overlapping with the EOT mode EOT signal of the nanohole array. And then we monitor the diffraction intensity changes by a CMOS camera. So what I did is, so here for example, we have a nanohole response denoted with the blue curve. The nanohole response immersed in water. And the laser response is spectrally matching with that. And this green one is the chip response in air. So, and here in this figure I have a fluidic nanohole chip integrated to a 2-channel microfluidic chamber. And now, so the 2-channel is filled with air, so EOT signal and the laser source does not match. So since we don't have any match, you see a black spot on the CMOS camera. So I will flush water in one of the chamber. So now what I'm doing is I am shifting this guy to a longer wavelength, so EOT signal allows this much photon pass through it. So you have a bright spot on the CMOS camera. So it's like an on and off. So when you have something on the chip, you see a light intensity change. So this is a simple example how we can use this phenomenon for biodetection. So for example, here we have again a plasmonic chip that is a response overlapping with the light source. That light source has been excited with a notch filter, and when something binds on the surface, here is protein AG plus IgG, it shifts toward longer wavelength, so light and EOT signal does not match. So when nothing on it, so you see a bright intensity. When it shifts toward longer wavelength due to the protein binding, you see nothing. So that shows that whatever stick on the surface, it's protein AG and IgG. So as I told you before, after I replace spectrometer with a CMOS camera, we can increase the throughput. So what I meant is, so here, for example, you see a CMOS image and a plasmonic chip which has very different, I don't know, like more than 100 sensing spots, and there are certain sensors with low intensity. This low intensity is due to the presence of the antibody in these particular sensors. So this figure shows that you can perform multiple detection of sensors all in the same platform. This light tracking phenomenon to determine the spectral shift in the EOT signal could be also used to make your own spectrometer with a camera. So what I used here, a liquid crystal filter. You can tune this liquid crystal filter with every 1 nanometer, and it has a 10 nanometer bandwidth. So I can create light source every 1 nanometer in the visible range. So then what I did is, so just try to remember this. This is the EOT signal, this black curve. When I take image with the CMOS camera for each wavelength, I see this light change, this light intensity change. And when you track this light intensity change, what you do is you recover that EOT signal by simply recovering these, by simply processing that intensity change taken by the CMOS camera. So here I'm showing you this spectrometer and our spectrometer results. They are showing similar spectral shift for different refractive indices of bulk solution and also protein AG IgG test. They're pretty much giving the same spectral shift in the EOT signal. So as I told you before, I performed a spectral integral method to increase the reliability of our platform by simply integrating this spectral integral or the transmission information within a spectral window of interest. Here we can perform the same phenomenon. Instead of integrating this transmission, we can simply determine the intensity, the intensity of the CMOS camera, which is the analogy of the intensity of the photons or the number of photons that reach to the CMOS camera. So using this, our imaging method and the spectrometer, we are showing very close limit of detection. Like this one is like 0.1 nanogram per mil slower, but it's very close to the commercial spectrometer platform. I will skip that part. And again, we can also integrate, we can also use this platform to real-time analysis of protein binding interactions. So here, the protein-protein interactions is done through two consecutive phases. In the association phase, you have a sensor, you coat the surface with a ligand that shows high affinity to your targeted analyte, and you flash a solution that contains targeted analyte A, and at the end of the association phase, you have an AB complex. In the disassociation phase, you flash a buffer solution to disassociate AB complex to give free A and B. And this whole process governed by the association and the disassociation constant. So if you look at this equation, you see that the spectral change in the EOT signal exponentially depends on this association and this association constant. And this figure shows that this spectral shift within the EOT signal while in the real-time experiment, while IgG binds on the AG-covered whole surface. So here I'm again showing you the spectrometer and our imaging-based platform, and they're giving very close exponential constant that shows that you can reliably use our imaging-based platform for the real-time binding experiments like people classically used in the commercial SPR platforms. And more importantly, since we are using a CMOS camera, we can perform the same experiment all in the same platform, but in the commercial SPR platform, you can only perform single experiment. So you have to repeat the experiment. It takes maybe like 3-4 hours experiment, and you have to repeat that. But in our platform, you can perform all the same experiment all in the same platform many times. So how many experiments you want to perform, you can define that much sensor spots on the same plasmonic chip. So at that point, I showed you how we can increase the sensitivity of the platform, make it simple in terms of optical readouts, and make it high throughput by integrating a camera. So after this point, I will show you how we can make the system low cost and portable. So what I did is I replaced this sophisticated and expensive optical tools like color filter, camera, source, and microscope with a very cheap CMOS camera and a LED source where the LED source is eliminating the plasmonic chip, and the change in the diffraction field intensity is monitored by the CMOS camera. So fabricating of the plasmonic chips are also expensive, so you have to make those chips as more affordable as possible. But classically people use e-beam lithography, but in the e-beam lithography you have to fabricate each sensor spot one by one, which takes hours and hours. But instead of using that lithography method, we introduced a new lithography method, which is called deep UV lithography. So what you do is you cover the surface with a mask and shine UV light source like for 30 seconds, and you fabricate tens of chips in a couple of hours. With this method, we can decrease the cost of the chip fabrication more than 20 times. And with the same optical quality. So again, the same principle here. We're monitoring the change in the light intensity. So here, for example, we have a detection sensor and a reference sensor. In the reference sensor, so when something binds on the surface, you see a light change, light intensity change. Since again we use a CMOS camera, we can perform the detection of different biomolecules on the same platform, which is called multiplexing. So here, for example, we have six sensors where 1, 3, and 5 are covered by protein BSA, and the rest is covered with protein bilayer composed of protein AG and IgG. I'm not sure you can see that, but the monolayer ones has a larger intensity, light intensity compared to the bilayer ones due to the more spectral shift in the bilayer one, making it less matching with the light source. It's same again. We can also perform a quantification of protein concentration by introducing different protein concentration. Here, for example, as you increase the protein concentration, you see a larger intensity change. You can also integrate a very low-cost microfluidic platform to this portable system to make a real-time protein binding kinetic analysis platform with a very low-cost and portable manner. So again, here as in that next presentation. So this is the commercial device result, and this is our handheld platform result. And when you look at the exponential constant, they are very close. So you can perform pretty much the same analysis in a very cheap manner, because here the cost was like $100. Here the SPR is like $100,000. And more importantly, you can actually that was the most important part. But the other important part is, since you're using a CMOS camera, you can perform multiple experiments all in the same platform, as opposed to the SPR analysis rely on one experiment. I will quickly show you also how we can use this platform to determine the sequence of the bases in a DNA template, which is called DNA sequencing. DNA sequencing is very important in many fields, such in biology, pharmacology, and determining the DNA sequence is very important to identify gene associated with disease, to develop personalized drugs, to monitor gene mutations for cancer research, and to understand gene functions. And the development of high throughput sequencing platforms, namely next generation sequencing, have been driven by high demand to the cost-effective sequencing. And next generation sequencing, with next generation sequencing, in one single step, you can determine millions of sequencing information. So in general, so this is the way performed by Illumina, which is dominating more than 95% of the whole market. They use fluorescent microscopes. So what they do is they label each base with a fluorescent tag, and then they have like 4 exams for each base, and like A, T, C, G, and which base gives the more signal, that is the right base in the DNA template. So they do this in many repetition, and then you complete the sequence in the DNA. But the problem, in the fluorescent microscope still exists, such as photobleaching, the number of mismatch between the bases and the fluorescent label, and also cost and preparation complexity of the fluorescent labeling. So instead of to address this problem, we try to get rid of the fluorescent stack in the DNA sequencing. So how we did this, we again used nanomolar arrays to real-time to determine the base sequence in a label-free manner. So what we did is we covered the gold surface with biotin-related dextran, then we introduced streptavidin. These guys strongly attached to each other, and then we introduced biotin-related template to the surface, which attached strongly to the streptavidin. So what you have is a DNA template on the surface that's strongly attached on the gold surface. Nothing can remove it from the surface. So then you have a solution containing polymerase and the bases, and the polymerase does this base pairing. So you have the base and you have the template. Polymerase take that base and stick to the DNAs, and then that DNA double-strand forks. So when you have the right base, polymerase does the correct base pairing, and then G sticks to C, and then DNA starts to elongate. So this is our setup. We have a manifold for the base selection and than washing steps, and we have a two channel microfluidic chamber. One is reserved for sensor, and the other one is control. And this is our real picture of our platform. And this is the spectral shift result, spectral integral result. Here the correct base is C because it gives larger spectral change compared to the other one. You see still some spectral variation for the wrong bases because polymerase still sticks on the surface, but the correct base, both polymerase and the base is on the surface. So you have larger biomass on the surface. So based on this evaluation, we determine the correct base, correct sequence in the DNA template with a very high signal to noise ratio. And more importantly here, so we got rid of the need for the fluorescence label, and also in the fluorescence label, many of you know, there is a background problem. You always have a background problem in the fluorescence microscopy, but here we have nothing in the background, there's all dark. You see just intensity coming from the light intensity, just coming from the sensor spot. The rest is all black, completely black. And we also integrate this camera to perform the similar phenomenon, like using control to monitor the intensity change to determine the sequence. You can also use this method to use in immunotherapy. I will quickly go over what is immunotherapy. So there are different ways of immunotherapy: the use of monoclonal antibodies, cancer vaccines, and adaptive cell transfer. So what they did is, and ACT is one of, I think, it's this most successful method. So this method depends on how well this T cell recognizes the tumor cell and kill it. So what they do is they take the T cells from the patient and isolate it, clone it, and then back infuse to the patient. So as I told you, this method relies on how well this T cell recognizes the cancer cell and kill it. So the recognition of the cancer cell by the T cell is through the activation occurs when the T cell receptor, TCR, specifically interact with the peptide major histocompatibility complex, or the PMHC of the antigen presenting cell or a tumor. So here we set up a platform how to characterize T cell, PMHC, and the PMHC and the TCR interaction. So what we did is we have an ATMA which is composed of a peptide. So in the edge of the peptide you have the PMHC molecule, and you introduce a solution to this platform. So actually I should briefly introduce that platform. So you have cells captured in a well, and then you introduce a stimuli which such that the peptide, the NTMA, released from the cell, and then the PMHC molecules disassociate from the cell membrane. So with this platform, we could characterize this imidazole, this PMHC recognition by the plasmonic sensor platform. So here we have two types of cells. Here the black one is the stained cell, which means the cell with the PMHC, and the green, the red one is the unstained one. I mean, these are the cells without the PMHC. So when we flush imidazole, then the cells started to secrete PMHC, and then you see a spectral shift. So this change, this slight change, is due to the change in the refractive index of the solution, because it was before RPMI, which is the cell media, and now it's RPMI plus imidazole. So when you delete this information from that, so you have a large change in spectral information in the stained cells compared to the non-stained cells. So you can use this platform to sort stained and unstained cells from each other. I mean, I don't know if you have time. If I will very fast go over this part. This is nothing to do with the optics, but I will show you what I have done in at MIT Koch Institute for Cancer Research. We developed a platform to weigh cell in a femtogram resolution. And we applied this platform to determine the therapeutic susceptibility of these particular cancer cells. I mean, you can use this platform to determine the susceptibility of many cancer cells, but for this project we all mainly focus on multiple myeloma. Sorry about that. Okay, so multiple myelomas are the cancer of the plasma cells that accumulates in the bone marrow, where they interfere with the production of the normal blood cells. Since these are the cancer version of the blood cell, they in the classical way they determine they detect this cancer by monitoring the antibody in the bloodstream, because these antibodies are secreted by the plasma cells. So if you have a tumorous plasma cells, you see an antibody that is abnormal for the immune system. So this is our platform. This is a cantilever platform. And the frequency of the cantilever change with the mass. So when a cell passes through the cantilever, the total weight of the cantilever change. So its oscillation frequency shift. And then we have a parallel series of 10 sensors. So each sensor weighs cell. So for the 20 minutes of period of 20 minutes, so within the 20 minutes, you can determine how, in a very high quality, how the cell's weight changes. I cannot also see that, but yeah, here also you can see this platform. So each cell passing through each passing through each cantilever, and its weight determined. And you have, so for example here, you have this is the data from each cantilever sensor. You determine this linear profile to determine how the cell mass change. And also we introduce a new terminology, which is MAR, mass accumulation rate. It shows you how fast the cell mass change. And then we map this mass accumulation rate per mass of each cell to have a single diagram so it's like a map showing you this cell with losing mass, increasing mass, all neutral. So here, for example, positive MAR shows you cell is gaining weight. This is losing weight, and these are pretty much show no change. So we use this phenomenon to determine how chemotherapy drugs affect the cancer cells. For example, here we have two same family cancer cells. We have a wild type, which is sensitive to that particular drug, and this is the resistant one. So what they do is, they produce this resistant cell line. So you have a population, you drug them, and you pick the one the one not dying. So you take them, clone them, and then make that resistant population. So when you look at that, when you drug the cells, the wild type starts to lose weight, which means they're dying, and you see almost negligible variation in the cell mass for the resistant one. This helps you to directly see the effect of the drugs on the cancer cells. So it helps the clinician to decide whether a particular therapy works for these cancer cells or not. So this shows that when you increase the incubation time, so here we drug the cell for 1 hour and increase from 1 hour to 5 hours, they die more. Here, the rest of one still show no response. We apply this different cancer cells here. I'm showing you again this MAR with a different profile. This is much easier to see. If you increase the incubation time with the drug, more cells started to die, and you determine this response just in a couple of hours. But in the classical way, these type of phenomenon usually determine like 3-4 months treatment. So the doctor gives the patients the drug and then let them go, and then after 2-3 months later they do blood tests. This is also another combination, drug combination. So here we have wild type and the resistant cell line, but here wild type is resistant to both. We have like the dexamethasone and Velcade. These are two well-known drugs, and this guy is sensitive to both of them. So drug 1, drug 2, it kills, but their combination kills them both more. For the resistant one, this guy is resistant to only this drug. So this drug kills it, this drug does not kill it, so their combination kills this much. This guy cannot affect on this resistant one. I will skip that part because this shows you another drug result. So this is the use of this platform for the real clinical sample. Here we have 9 patient sample we directly got from after the biopsy and put them in that cantilever platform, and we see that we can successively group those sensitive, resistant, and negative. Here the negative are the healthy patient. We can successfully group those patients for in terms of their drug response. And again, as I told you, this platform could help the clinician to decide the best drug or drug combinations within like a couple of hours of measurement. So as I told you before, in the clinical studies, what they do is they give the drug to the patient. That patient goes and they come back, and then they look at kappa LFC. This is antibody, so they check the antibody level. So for example, here our platform says this guy is sensitive. This guy is sensitive by just looking at the MAR profile, because the MAR is going negative and negative with the drugs. But if you look at these drug treatment, the classical drug treatment could say that after 30 days. So this gives you a lot of time, because multiple myeloma is this number 2 in death, the cancer patient death, and you have at most 7 months to save that patient's life. So even 1 day is very important. So, and if you consider like 90 days period, maybe you're almost too late to save that person's life. And actually I would like to share you one that I think it was number 7, patient 7. So this was my 3rd experiment, and I was keep doing the experiment over and over again. I see nothing in my platform, and I called the doctor two weeks later, and I asked her, okay, my cantilever doesn't respond anything. I have a problem. She said, no, we were expecting, because she was so resistant to any drug combination, and that patient died. So it's that critical. And one last slide. So I did many. I developed different types of optical and electrical based platforms, but now I started to I would like to commercialize this platform. So So I have the idea, we had a team now, we kind of found the funding. This is the most important part, otherwise you can, I don't think you can do anything. I need money with some intelligence, it's like intelligent person. And then if you have the money, you should have some prototype, and then this period actually shouldn't go very slow because then this guy will hate you. So then after you did the prototype, you have to validate, you take your system and then deliver to different labs and get some feedbacks, and then if they say okay, or if they don't, if say you should improve that part, and then you improve those parts, give them back, and then if they validate, then you launch it. So in my lab, we started to, we would like to commercialize our platform, and then we have our startup Bioscope for different technologies based on mainly portable sensing platforms. This is our first prototype. I'm using a TorLab XYZ stage, but we're not using them because they're super expensive. I can maybe manufacture 10 microscopes with the cost of one XYZ stage from TorLab. So this platform can be used for different labs like pathology, histopathology, if people doing cancer research or any type of like basic research. So this is like, we could get even like 1000x image, like these are all like single cell. This is like 200, so you can easily recognize cells in the whole picture. And now it's all this product is in the beta test right now. We're talking with people, and we would like to commercialize this product. So in the future, if you're looking for a job, so